At the University of Bristol, a new life sciences building is currently being constructed, and Shirin and I were involved in coming up with ideas for an art installation of large glass spheres with biological things inside. After having come up with several initial ideas, we tested how viable these were, with varying degrees of success. Using agar jelly and different types of algae, Microcystis and Planktothrix, we hope to coat the inside of a glass container with greens and reds. Working in the lab with a net, we coated the insides of glass containers, swirling the heated up liquid agar around the sides. After adding some of the algae culture and leaving for a few weeks, the algae appeared to have grown, however not really up the sides very much. Another attempt involved mixing the algae with the agar, and a third involved waiting for the agar to dry and then inoculating with a much stronger culture. This was far more successful and would potentially look very effective in a large sphere. After doing this, we thought of several other possible variations we could try. One was to grow plates with strong coloured cultures of algae and suspend these in a mobile-like arrangement inside the glass sphere. Another variation was Annette's idea, in that she had quite a few test tubes which had already been used for algae cultures. However, once the culture had been removed, a small line of dried pigment remained. We decided these could be sealed and then hung up in the spheres. We sealed them by going to the glass blower's workshop. Also, the fact that they are not reliant on nutrients and could easily be changed, as Annette often has quite a good supply of them, meant that these could be a simple, effective solution. Next were the mushrooms. We bought the pink oyster mushrooms to test out, and we followed the instructions, which involved filling the plastic bag of hay with boiling water. Unfortunately, we had rather a small kettle. Then the water was allowed to soak in, and we then added the spawn. I also wanted to try the spawn on some plates, as I thought it might be more attractive in a glass sphere to be on gel, so we tried this as well and put the plates into the same environment as the mushroom bag. After about a week, there was not a huge amount of growth. However, the bag mushrooms had grown dramatically in 2 weeks, and 2 weeks after that, even those grown on agar had developed. We did use 2 different types of agar, BG11 multi-nutrient agar and chlorella soil extract. However, both seemed to be successful. This idea could potentially be quite successful. However, it would involve keeping the fungi environment quite carefully monitored. There are many very interesting different species, but these may be difficult to grow and get our hands on, so potentially a more difficult reality. The moss balls turned out to be rather successful. Once the motor had been placed in a position that meant the water from the bottom was constantly being pushed upwards, so the moss balls didn't get stuck at the bottom, it allowed movement whenever the sun was out. The sun, therefore, was the greatest limiting factor. The Selaginella didn't really require much more research, as it seemed perfectly doable. The only issue would be that it may not look attractive, as it would go brown and start to decay after some time. We looked at iridescence by going to the zoo to gain inspiration, and made our own models out of modrock and paint. Shirin created an iridescent beetle, and I attempted a radiolarian and a coccolithophore. As these are microscopic, therefore we would not be able to have life-size forms anyway. These obviously don't have any biological constraints, so could easily last for a lengthy period of time. We then looked at a variety of climbing plants, both at the zoo and the botanical gardens. We decided this would also be quite a viable option, as no doubt the life sciences building will be heated, so relatively tropical or at least Mediterranean plants would survive. The main issue would be preventing them from completely taking over the space. We were also given a test climbing plant, Tradescantia, to see how long a cutting would take to start growing. Within 3 weeks the plant was growing up towards the light. so it would not take long before it started looking quite impressive. The Vinogradsky columns were in fact very easy to do, as the second year biologists were studying the different contents of the River Yeo and other locations. This meant that we just needed to monitor them and document the changes. The final pictures we took of them showed really quite an impressive range of colours, and with light shining through them it would look quite effective. The main difficulty would be that if a glass sphere was to be used, they may be rather heavy, however some method of supporting them could be used. The last idea we had involved using ants to burrow into gel that provided them with nutrition, thereby forming tunnels. Unfortunately, the ants decided against eating the gel, so we did not end up with any tunnels at all. If this had worked, it would not need very much maintenance and would only need changing perhaps once every 4 months. After having tried out these ideas, we managed to find several which could definitely be effectively utilised. We had help from many people and were very lucky to be able to have been given resources and time. Despite a change in direction with what is going to be installed into the Life Sciences building, the project has been successful in creating links between arts and science, and both Shirin and I have had an invaluable experience being part of it. So thanks very much to Heather Whitney, Steve, Annette, Tom Pittman and everyone else who helped us on this project. We've had a great time.